Let me first thank the organizers, MPOC, for inviting me here for this lecture on dietary fats. And Dr. Sundram is, in fact, a co-investigator of this collaborative project between us and MPOC. Because it's, I'm going to start with a bit of trivia because it's too early for hard science. So let me tell you about the history of fatty acids. That was started way back in the 17th century, and it was the German alchemist, oil chemist, so to speak, Otto Tahenius, is the first, was a description. In this book, he described what we now call a fatty acid. He identified as an acidic substance. And the composition wasn't actually detailed till the 19th century. I acknowledge Prof. Murphy, who was the, actually described this in a very elegant talk a few years ago. In the French chemist, Marche Mouret, I don't know how I pronounce it correctly, he's the one who introduced what we call the margarine. And even those days, there's a business development and commercialization angle to this, because even those days, the government has to, the industry has to respond to the government request. So it is claimed that his work followed a call by Napoleon for an alternative to butter. And although this is sort of claimed to be to promote the well-being of the working class, more pragmatic reason, motive has been to fight the war in Prussia. However, margarine didn't help the war. They lost the war, right? So, and fatty acid became big industry. And then it was bought by a larger company, which currently is the Unilever Group. And now get into the, so that margarine from discovery to the current state of art has gone through a lot of phases in the oils and fats industry. For commercial food application compared to the edible oil we see on the shelf, if you like, liquid oil, most commercial food applications need a harder fat. So that is achieved by blending, fractionation, or hydrogenation up until recently, because that produced the desired fat in a way that the industry demand. Then as the years gone by, we realized that trans fatty acid is not that good. And so there is a move to be away from generating a lot of trans fatty acid for commercial foods. What happened in the case of a hydrogenation, if you start with in this case, oleic acid, which has a melting point about 4, once you hydrogenate and get rid of this double bond, it's simply the melting point now is about 46. And basically, it's the kink disappears, and it becomes a saturated fat and behaves similar or worse than a saturated fat. So zero trans fat is a recent market entry compared to relatively. So it's achieved by interesterification, either by chemically or enzymatically. Enzymatic pathway used to be a lot dearer, but I think with the advent of new enzymes, it is becoming more affordable. So as previous speaker Tilaka mentioned, it modifies the triglyceride structure within the native oil. So it ends up as a randomized fat distribution, and the health outcome is not fully established. And Tilaka covered it elegantly, the aspects of endothelial dysfunction, which I believe very dear to my heart, and because the current theory is the vascular inflammation leading from the generation of inflammation will influence that endothelium, and even a little bit of cholesterol which now has the capacity to go a longer way because your barrier is somewhat compromised. So there is some reports that IE fats does not lower cholesterol, but as we know, the current knowledge base is there are a lot of mechanisms beyond LDL cholesterol that can promote vascular inflammation and the pathogenesis of cardiovascular disease. So it is not only cholesterol, there are other things happening in the background. So so this is a interesterification thing. This was achieved by blending. This is a slide from Novozyme, one of those enzyme manufacturers, and you can see it is free of trans fatty acids. So the objective and aim of this present study was to evaluate the effect of different fatty acids in the presence of a comparable intake of linoleic acid. And those endpoints are what we were interested in is the sudden cardiac death and susceptible to cardiac arrhythmia. This is a rat model because we, as I mentioned in yesterday's talk, these endpoints can't be easily achieved in human population. So just to understand whether there is an influence on these, so we decided on that. So in particular, we want to compare several commercially important fats and oils with particular emphasis on the impact of stearic acid, interesterified form, against a naturally occurring palm oil. So these are the fatty acid composition of the test oils we used. We had great difficulty, even with all the contacts Dr. Sundram has in the oils and fats industry, to come up with a HSIE, high stearic interesterified fat, with relatively rich in stearic acid, because if you look at its 18.2 content, this is more than what we wanted to have, because we, as I said earlier, we wanted to match the 18.2 intake, but we had to sort of balance the diet against that high 18.2 background in the high stearic acid. So we were not quite happy with the design from the first day onwards, but we had no alternative but to carry forward. So these are the blends. We came up with blending different oils and fats, primarily palm oil and sunflower oil, to achieve 18.2 intake, so that we match the 18.2 intake of commercial fats and oils, and we always use a positive control in our experimental design. In this case, the proven antiarrhythmic actions of fish oil, that we use fish oil as that. You see fish oil is lower in 18.2, but it is PUFA comes primarily from the omega-3 fatty acid, which is about 30% in our case. So from energy point of view, you can see they are pretty similar, and from 18.2 derived, the energy level is as so forth. And there were no differences in the growth and development of these fats on the growth rate or the final body weight, so they all is very well accepted by the animals and went forth. Then we went to look at the body fat distribution of the compositional studies using DEXA. This is usually used in the human, but we have another model that will be ready for small rodent models. So there was so any dietary intervention can be examined using this thing. So here again, this is the pre-diet values. Compared to those, the end of feeding, there were no differences in any of those. And this is the technique I described yesterday. What we try to do here is we pre-feed the animal, and we put a ligature here and induce if you like, a myocardial ischemia, because the blood wouldn't go below that. And we monitor whatever the arrhythmias, that means the aberrant rhythm, moving away from a normal rhythm to a different increased rate or no rhythm at all, which is called ventricular fibrillation, within that 30 minutes period. So we count and grade them, and there is an internationally accepted way of scoring these, and we adhere to those and come up with a score to suggest how bad or how good it is. So if you look at the ventricular tachycardia incidence, it's 20 out of 20 animals in the high stearic group developed cardiac arrhythmia, and it was slightly lower in the palm oil, and fish oil also developed high, because ventricular tachycardia is very easily developed upon a disturbance in blood flow. So when you touch an animal in the heart, it's what's first abnormality you see is what we call ectopic beats. The ventricle normally has a beat pattern, but once you get a little bit of disturbance, they develop an ectopic beat, and they get deteriorated into a tachycardia. And then if it's been fed a good diet, the tachycardia is less, or it will not develop into a fibrillation or less fibrillation. So the incidence of VF, again, similar, except for the fish oil group, which showed its well-known protection. So the duration-wise, this is the same, and we was lowest in the fish oil group. The other HSIE and PO were not different. The, again, this is the number of, the number of animals who develop VF, that is 16 animals out of 20 develop an EVF. What I mean by a VF death is that those animals who had a VF acidose, in this case there were 16, and 13 of them died, it was fatal. In the case of rat has a different collateral circulation system, unlike humans, because if we get a VF, that means end of us, unless you get a defibrillator and someone knows how to use it, or mouth-to-mouth resuscitation. That's why most of the US airports got a DC defibrillator within 100 meters of each other. And so this is also an indicator of how, if you like, vulnerability can be, because if you have a more time with a VF not going straight into fibrillation, but there is a bit of hope, so there's the recovery can be quick. So we can also calculate the so-called an index of VF episodes during the 30-minute period. You can see there were not much of a difference, no statistical significance, but these three tended to be a little bit higher. So average survival time was greatest in the fish oil group, and this HSIE palm used to be lower, but across group comparisons was not clearly significant. The total mortality was again lowest in the fish oil, and the high oleic sunflower, which is about 84% monounsaturated fatty acid, was also protective. There was no difference between the stearic and the palm. We also did the fatty acid incorporation into tissues, and you can see there was not much of a difference across the fatty acid incorporation into different tissues, because we remember that we balance 18-2. 18-2 tend to dictate most of this tissue distribution of fatty acid, because the animals will try to maintain its homeostasis. So if you give sufficient amount of linoleic acid, so we are not really surprised by that. In fish oil, on the other hand, the body has a craving for long-chain omega-3 fatty acids because the body is deficient. So even a low amount of fish oil, even if you put 5%, 0.5% rather, it will get incorporated very efficiently. So because, as you know, most of the brain, neural tissue, that's where the concentration of omega-3s is. So, and heart, every excitable tissue has a significant amount of long-chain omega-3s. It's not omega-3s because linolenic acid is also omega-3, but it does not share the same benefits as the longer chain, 20 carbon atoms or longer. So the fatty acid incorporation weren't that different. So what was the reason for we are probably not seeing an effect? Because if you go back and look at the final blends, we can see, because we had to dilute out those 18-2, both HSIE and PALM had very similar palmitic acid content. So in a way, we not been able to compare the 18-0 versus 60-no effect, because predominantly these are quite similar. So the 18, although this is probably not sufficient, there's a 12%, because in the commercial situation, the 18-0 would be a lot higher. So that's probably the reason that we did not see. So I want to shift the gears into something I mentioned yesterday again, the matrix interaction. It is important to realize that we just don't eat fat by fat itself. There is something else accompanying it, which is primarily a carbohydrate. And if you walk out to the streets, this is all refined carbohydrates. I mean, the bakeries and most of the places, including fructose in the US diet, it is being now blamed as the biggest problem in relation to obesity, soda to everywhere. So a given carbohydrate or a given fat may behave differently depending on what is along with. So if you feed even a refined carbohydrate with a good fat, the outcome may not be the same. right? But in alone, isolation is one thing, but when you combine into our plate, the physiological outcome might be different. So that takes me into a situation in my native homeland in Sri Lanka. Because Sri Lanka population is you may not believe it, is the second highest internationally for vascular disease development. Yet their cholesterol is normal, right? So below the accepted level for cardiovascular disease. So what I proposed way back in 2003 is because it has been shifted to a very high carbohydrate high glycemic index, highly digestible carbohydrate. And Sri Lankan diet so far, until recently the introduction of palm oil to the diet, has been largely on coconut oil. And coconut oil does not have sufficient 18-2. And if you look at the cross-section of Sri Lankan population, they suffer from borderline or frank EFA deficiency as a reduced intake of 18-2. Under those circumstances, it is very evident, at least in my eyes, that it will promote the inflammatory pathways if you look at the Sri Lankan population, their high triglyceride synthesis occurs because high TG, HDL, low HDL is the common thing, and they are marginally or borderline hypertensive, about quite a bit of people. The treated hypertensive, that is, there are a lot of undiagnosed ones, and this all can result into endothelial dysfunction. Again, with that, we looked into can there be an interaction under those settings. The effect of 18-2 is quite important. Even Dr. Khosla, who is in the audience, some time back shown elegantly with Casey Hayes that 18-2 component in the background diet is actually important in the outcome of a given saturated fat. So with that in mind, what we did was we used some coconut oil and fed them for some time as before, and then ligated. You can see coconut oil is very proarrhythmic under that setting, right? Then compared to lard, so what then we did was, these were done on cornstarch, is the background starch. We replaced with a complex carbohydrate called high maize. And we have a proprietary carbohydrate resistant starch, and this is another one that you can commercially available. And what the complex carbohydrates do is they escape the digestion in the upper gut and they travels down. So it's low glycemic, released over time, and more importantly, it is being chewed up by the gut microbials to generate good fatty acid. There are the short chain fatty acid, butyrate, propionate, and acetate. The importance of gut microbes is huge, like it has only been recognized in the past few years. I have a couple of slides on that. Now, it is known that cardiovascular disease development may in fact start in the gut, because the microbial metabolites generate different toxic substances which leading into inflammation. So, what we saw is when the bad actions of coconut oil was in a way positively influenced by having a different carbohydrate. So in the case of VT, VF, so this lard was 58, so 87, but when you put coconut with a different carbohydrate, we were able to reduce that. The arrhythmia score is the overall parameter you can calculate by those individual areas, and we saw that is quite influential. So this is pretty strong and important stuff. I'm going to quickly show this unpublished. We just finished recently data. Again, that concept, I showed this one before. You can see if you this was the surprising aspect for us. We compared high oleic sunflower and milk fat in the presence and absence of fructose being a simple carbohydrate. And what we're seeing is, in the monounsaturated diet, fructose incorporation has increased blood pressure. So there are things, although on the face value from other endpoints might be good, but when you put it in the context, they are behaving differently. In contrast, the milk fat had no effect, and also I can tell this audience, this was finished last week, so we compared the refined RBD palm oil and also with the caroteno products with the high red palm oil. there was no difference in interaction with fructose. So I think we may be into something. We want to develop this further. When we measure the body composition, there is a trend in the HOSO diet to go up. But this was that caught my eye last week, because if you had that red palm oil, there was a reduction in the percent fat mass as measured by DEXA. Whether the antioxidant component has an effect on that in the presence of a high fructose, I don't know. I have no explanation, but just an observation as of last week. So in summary, ladies and gentlemen, in that particular experiment on the HSIE, there were no difference in the growth rates. And fish oil, as a positive control, had the greatest protection against cardiac arrhythmia. The arrhythmia outcomes were similar between the three saturated base diets. But a limitation of the study, as I alluded to earlier, is the fact that higher stearic, we couldn't maintain the stearic acid levels to say this is a good comparison between 16 apples to apples comparison, so that couldn't be achieved. Hopefully we'll be able to get a more purified or a more enriched 18-0 preparation for our next series of studies. So again, the high level of linoleic acid in the fat sample is required dilution with palm oil. So this, in the end of the blending, palmitic was very similar. There is another thing is about balancing for 18-2 intake. There are I think the scientists are divided. Some people say dietary fats should be tested in their own right. So if it's a coconut oil diet, you feed coconut oil, and noting it's avoiding EFA deficiency. When you try to bring in the, then they say that that's the only way. And if it's a palmitic acid diet, you should feed palm oil rather than trying to balance the 18-2 intake. There is other group of thoughts say, no, you always must allow adequate intake of 18-2, but it might have its deficiencies from the fatty acid wise, or because 18-2 is one of the essential fatty acid the body requires. The body can't synthesize that. So we need to rethink of how we approach dietary fat related experiment, at least in animal models. So there is we couldn't actually make a proper conclusion by comparing these two. So I just want to introduce a couple of slides about the current thinking of the gut microbes in CVD development, because there is a clear set of evidence that microbiota, because everyone carries a different amount of, a different profile of bacteria in the gut, and everyone seems to vary from a given stimulus, their outcome. This has been underestimated, these guys, what they do in relation to human physiology, and now we know from IBD to inflammation and CVD is actually dependent by the microbial population in the brain. There's an excellent paper by the Swedish group in Nature that I recommend if you want to get into know more about how important the gut role play in the chronic diseases. That is a must. And if you look at the metabolic capacity of the gut microbiota, they promote adiposity, they influence that metabolic process in peripheral organs, including the brain, satiety linkage, and gut hormone release, and synthesis, storage, and metabolism in the adipose tissue, liver, and muscle. And the last point is the one. So the major constituent in the diet in the microbial outcomes may be different. In fact, we started looking at different fat and matrix interaction with regard to those outcomes in the short chain fatty acids. I can tell you, bit of privileged information, that polyunsaturated fat of commercial interest cause a detrimental outcome compared to your saturated fat. So that's unpublished data, but so this field going to grow. There's no question about it. That's because if you look at the diet, the microbialism, the host metabolism, these are the independent effects. Those are the, if you like, the external stimulus independent of the diet. But the dependent effects, the Stanley Hazen from Cleveland Clinic has been the pioneer, which caused quite a bit of controversy 2 months ago when he said the meat-rich diet causes abnormalities in that simply because of the bacterial population, right? That is coming from the TMAO products in there. So you can see the choline component of the phosphatidylcholine head groups, and those present in Coca-Cola can actually carry sufficient carnitine is the other culprit, causes an abnormality in the bacterial metabolism and ending up inflammation in the vascular and the cardiovascular disease development. That is his hypothesis. He got a very excellent data set. And then again, all these dietary difference of material, including, I put my $10, including type of fat, will influence the outcome of gut bacterial metabolism, leading into disease development and so forth. And that probably explains the variation in a human clinical trial that we observe to a given stimulus, because every individual there are responders and non-responders. We've done a lot of gene sequencing, it's not me, but the group working on microbial, CSIRO, and they can separate the people with very classic relating to the microbial genomics. So it is a growing area. I think it should be, and another angle, the edible oil industry, in my opinion, should give some thought and to put some money, because it doesn't seem to be a very simple cholesterol-related outcome. And with that, I think I'll wrap up the thing, and thank you for your attention.